Okay, welcome back to part two of chapter 32 on an overview of animal diversity. So we stopped at talking about the four geologic eras of animal species diversity, and we watched a video giving you a good glimpse of how what it would be like if everything did happen according to what science predicted. So, what other similarities do animals share? So, this last big section of the chapter focuses on body plans of animals. So, we shouldn't take up another hour for the latter, this second part, but do expect that the next two chapters will be pretty long because they do have a lot of species to go over, just like we have talked about with plants. And we're going to go on and talk about the different animal species in the next two chapters. All right. So, what are these body plans? So, animal species they found that have major body plans that are similar, and they are particular set of morphological and developmental traits that are integrated into a functional whole. That's what science means by having body plans. So, body plans, in a sense, provide a way for us to compare and contrast key animal features that we can see in different phyla or species of animals. First of those being symmetry. So I kind of briefly talked about this earlier. You have different symmetry with some animals being bilateral symmetry, some animals being radial symmetry, some don't even have a symmetry at all. So what are these different symmetries? Let's start with talking about radial symmetry. Radial symmetry are when body parts are arranged around a single main axis that pass through the center of the animal, kind of like you think about a cylinder looking thing, and here's that central axis, and so the animal kind of arrange themselves around like a ring around the central axis. So with radial symmetry, any slice through the central axis will always divide the animal into mirror images or equal parts. So again, imagine a perfect circle cylinder and you're slicing through in any way, whether it be horizontal, vertical, or diagonals, you will have mirror images. And what do we mean by that is exactly in this image here, showing you the example of Hydra. Hydra is a simple form of animal where it has a single opening that serves as both the mouth and the anus and is surrounded by circular rim tentacles like this at the top. They usually capture their prey with their little tentacles and then they push it into their body cavity, which is this interior hollow space here that is formed by your blastocele, and then the gastrulation process forms the archenteron. But we'll talk about that in a little bit in another section, but that is how it has this hollow body for digestive purposes, and these tentacles will push the things inside it to itself. That's how Hydra looks like. And so, if you were to cut it this way or this way down the animal, you will still have mirror images of the animal itself. And then, now, most sponges actually have irregular shape, kind of like this. Like that. Is not, rarely do we have sponges that are perfectly symmetrical, but not saying that there aren't. So there is a specific sponge species called barrel sponge, where it literally looks like a barrel like this. So they are arranged in a regular pattern around a central axis. So that's why they still have what's called the radial symmetry. So Hydra is a species with radial symmetry. Barrel sponges is another example of species that have radial symmetry. We'll talk a lot more about these different invertebrates that have these different symmetries in the next chapter. But know that most sponges are kind of irregular like this, so they usually is in the category of absence of symmetry. Second type of symmetry is bilateral symmetry. This is when body part is arranged around two axes of orientation, a head-tail axis and a dorsal-ventral axis. Dorsal-ventral is an anatomical term where dorsal means the back of the body and ventral means the front of the body. If you're talking about bipedal organism, meaning they walk on two foot, like an upright human being. But for animals that are on their all fours try on their all fours, like monkeys, dogs, etc., then the dorsal will still be their back, but the ventral side, we're talking about their tummy side. I always draw this little dog or cat, or whatever it is. It's a four-legged animal. Okay, I can draw, but I'm just showing you a really quick image. So dorsal is then the back side, and then this is their anterior side, and then if you look at the on the side like that, okay, this is their back side, dorsal side. This is the ventral side for a four-legged animal. So only one slice can divide the animal into mirror image halves when we're talking about bilateral symmetry. So that's why it's called bilateral. They have two mirror images that are bisected by one slice down the animal. Okay, and they will divide the animal into the left and right side. So for example, you have a lobster right here and an owl right here. So either you have animals on all fours or animals that are upright like this. You still only have one slice that can happen down the center of the animal and divide it into left and right halves. That's what we call bilateral symmetry. Lobsters, owls have bilateral symmetry. And usually, eyes and other sensory organs are concentrated at the head end of the body versus the tail end. Of course, there's always exceptions, but most of them do have eyes and, mouth, nose, eating things around their head area. All right. So animal symmetry usually generally fits the animal's lifestyle, going back to why it's a natural selection kind of thing that drive these formation of symmetries of these animal species. So radial animals are usually what's called sessile, or you can think of them as being living attached to a substrate. They don't walk around, roam around. They're attached to this particular area. Or they can be planktonic, which is, think about what planktons do. They are drifting or weakly swimming in the ocean. So radial symmetry animals are usually either you're not moving at all, non-mobile, sessile, or you're planktonic, slowly drifting around. In this particular case, you have sponges that are attached to a rock. They don't move around. They're sessile. So why radial symmetry fits their lifestyle is because they don't move. So having radial symmetry helps with meeting the environment equally well from all sides. Because you can't move, you better have things that can absorb nutrients or meet whatever environment surround the surrounding environments, such that all sides can have equal chances of colliding or diffusing nutrients into the body. So that's why radial symmetry is meant for animals that can't move or hardly move at all. Now, bilateral animals usually move actively from place to place. So, usually sensory equipment, that's why they're concentrated at the head end of the body, and it usually includes a central nervous system. Central nervous system has to do with your brain and spinal cord for enabling coordination of complex movement, because bilateral animals have a lot of more complex movement compared to radial symmetry animals. For example, if you imagine a dog, there's crawling, burrowing, of course, or animals and that flies like birds. They have to coordinate their wings a specific way to fly properly. And then for animals that swim, they have to coordinate their body in a different way. Even if we go back to the dog's case, they need to know how to move their body in order to swim, stay afloat, and also versus crawling, digging into the dirt. So again, this is central nervous system is another very interesting topic in AMP1, where we talk about the cerebrum and cerebellum and how those two are parts are the brain parts, and how the cerebellum records movement patterns, such as so that we don't have to consciously think about how to crawl or walk or swim anymore once we have that pattern memorized and set in. So these activities, in a sense, become second nature faster with repetition due to having a central nervous system, such as the brain. Within the brain, it is our cerebellum that records those movement patterns that we have developed and repetitively go through, so we don't have to think about these movements anymore. But that's why bilateral animals have bilateral symmetries, because they have to move from place to place. They have to have sensory areas in the head so you know where you're going, you see where you're going, and you feel you bump into something, you can turn around, etc. Okay, second body plan that we share amongst all animals is tissues. Embryos usually become layered during gastrulation, as we have talked about. So diploblasts are those that have two germ layers. Remember, we talked about how from a blastula, you have this hollow blastocele within there. Cell starts migrating into there. So here's a hollow ball with the blastocele hollow space. And then you have this migrating of cells towards the center like that, migrating into the blastocele. This is what we call gastrulation. So that's how we form a outer layer and an inner layer like that. So that's why those embryos or animal species that have embryos with two germ layers, the ectoderm and the endoderm, are called diploblasts. One example of those animals are cnidarians, which is jellyfishes, for example. So ectoderm is the outer layer that you're seeing there on the embryo, and it covers surface of the embryo and usually develop into the outer covering of the animal, like your integumentary system for humans. And we're talking about humans, which is your skin. Dogs have to have fur, skin, etc. And sometimes in some animals, it is also directing for formation of the central nervous system, or the CNS. Um, Endoderm, on the other hand, is the inner layer that's formed by this indentation of gastrulation. That inner layer is called the endoderm, which is the innermost layer that lines a pouch formed during gastrulation. So there's that pouch right there within. So this is the endoderm, and it surrounds a hollow pouch, again calling us, as a hollow space called the archenteron. Archenteron is what will ultimately develop into the lining of your digestive tract or digestive cavity, and then the lining of organs as well, such as your liver and lungs. So triploblasts, on the other hand, will have three germ layers. All triploblasts are bilaterally symmetrical animals. For example, vertebrates like us, human beings, have three germ layers. So ectoderm and endoderm still form the same stuff like the diploblasts do, but they have a third layer called the mesoderm that fills in the space between the ectoderm and the endoderm, and usually they will develop into muscles and organs between the digestive tract and outer covering of the animal. So anything from deep within your digestive gut tract, stomach, intestine, between those organs and whatever is between those organs and the outer covering, such as your skin. All of those connective tissue, for example, is then made possible by your mesoderm. All right. So gastrulation, I already said, is a process where a group of cells will move into the blastocele of the blastula and form a tube called archenteron, which will form the primitive gut, which will reach the embryo surface with an open end called a blastopore. So here's the whole process here in this picture. Here's a zygote, and from the outside, that's all you're seeing. You can't see what's happening inside, which is why they kind of do a cross section of the embryo. So you see what's happening on the inside. Here's your hollow space, hollow ball called the blastula stage with the blastocele, that hollow space in the middle. And then you start to have indentation of your outer layer of cells, start to form what's called the archenteron. And now you have an opening down here called the blastopore. When the gastrulation is complete, you have this completely empty space right here called the primitive gut. And this is now the gastrula stage of the embryonic development process. Now, gastrulation also forms your primary cell layers of animal body, which is either two or three layers, the ectoderm, endoderm, and mesoderm. In diploblasts, blastopore at the bottom, which is right there at the bottom, will becomes open end of your gastrovascular cavity. So here's diploblast. For example, in the Hydra's case, you have this ectoderm on the outside, endoderm as yellow, slowly migrating upwards until you reach the top. And that's how you form this whole hydra with an opening at the top as your vascular activity opening to your gastrointestinal tract. Yellow layer is the endoderm, blue layer is the ectoderm. Now for trypoblast, there's um, two ways of development. That's why you have something called protostomes or deuteriostome. We'll get more about that when we get to another slide, but for now, just know that there are different ways of development. So trypoblast protostome is where the blastopore will become the mouth. So here's the ectoderm, endoderm, and then all of a sudden you see these red cells that start to bud off from the endoderm. That's your mesoderm cells. Mesoderm cells here, eventually filling in the space between your ectoderm and your endoderm. And here's the blastopore at the bottom that will eventually become the mouth, for example, in an adult worm organism, animal body. That's where those layers come from, form or where those what those layers form. And then triploblasts, deuterostomes, such as sea urchin, you still again have something similar as your ectoderm, your endoderm, and then here's your budding of your mesoderm cells. And mesoderm kind of form things on the outside, but there's your ectoderm forming this digestive tract. And mouth actually forms the opposite side of the blastopore, where this open end becomes the mouth, this open end becomes the anus, for example. So that's why you have the mouth on the top and anus at the bottom. And that's different ways of development between two triploblasts, whether you're triploblast protostomes, or triploblast deuterostomes. Body cavities is the third thing we share as animals according to our body plans. What are body cavities? They're a fluid and air-filled space between your digestive tract and outer body wall. Any space you have between those two areas, they form body cavities. So all animals have a body cavity that is usually between your endoderm and your ectoderm and provide structural supports. They serve very important functions. They also facilitate internal transport, such as nutrients and gases and wastes. So this is like your vascular system, for example. Um, that will be inside your body cavity that allows for, let's say blood vessels to transport those nutrients, gases to your lungs, and then waste towards your either your urinary system or the bottom of your digestive system. So triploblastic animals have what's called a coelom, which is a body cavity formed from the mesoderm itself. And those that have a complete coelom, we call them coelomates, as another term for these triploblastic cells with a coelom. And it is that coelom is what form layers to suspend internal organs, because you have this huge hole, in a sense. That's what cavity is a open space for all the organs to fit within there. And so when you have all these organs in there, you got to somehow have some way to suspend them so they don't, disorient themselves or mislocate to another location of your body. So in that case, that's why you have a way to suspend the internal organs by the coelom. And then there's sometimes fluid that cushions your suspended organs, because they're usually these internal organs are very delicate. You want to prevent them from any internal injury. If you happen to have a blunt trauma to your body, or animal faces a blunt trauma, falls or is hit by something, in earthworms, for example, as you can see here, the coelom fluid acts like a skeleton against muscles because they don't really have visual bones. So what acts as the skeleton is actually the coelom fluid that gives them movement with their muscles. Now, they also enable internal organs to grow and move independently of your outer body wall. What do we mean by that? Well, without your coelom, beating of your heart and ripple of your intestine will warp your body surface every time they move. Like imagine if you don't have any body cavity that surrounds your heart, there's actually a pericardial cavity that allows your heart to be sitting in that particular location. So every time it beats, you might actually see that your skin moving around that area, indentation and protrusion as your heart is moving, or when your intestine is moving, your abdomen can be moving. But all of that is kind of contained within your body cavity and by your coelom, such that they don't warp your body surface, change your body surface, and rippling that you will be able to see if they don't we don't have them. All right, so some triple blasted animals also have, instead of a coelom, they have a hemocoel, which is a body cavity that grows between your mesoderm and endoderm. So here's your ectoderm, here's your endoderm. So the red layer there is then your mesoderm. So for those animals with a hemocoel, that particular area, that hollow space you see, usually contain what's called hemolymph, which is a fluid that functions to transport nutrients and waste, just like your blood. That's why it's called hemolymph. Hemo is kind of like your blood, stands for blood as a prefix. Lymph is the lymph fluid that goes around as you're part of your immune system. And some triple blasts, lastly, don't even have a body cavity at all. So they usually tend to have thin, flat bodies. That's why they don't have any body cavity and they don't need one because they're flat. So they don't need any internal transport system to let things go around their body. They just allow diffusion to happen because they're so thin and flat. Diffusion can easily cross a short distance, efficient enough to supply the animal with whatever they need. And so these animals are sometimes called acoelomates, which means A means no for biology terms a lot of times, coelo for that hollow space, body cavity, mates, the animals. So these are animals without a coelom is really what it is. All right, fourth thing that we share with animals in terms of body plans is that protostome and deuterostome development that I talked about earlier. These are developed modes distinguished by the differences in cleavage, coelom formation, and also the fate of the blastopore. So that's why there's these two distinguished ways of development. First, let's talk about how they differ in cleavage. Remember, cleavage is the even cell division that happens after after a sperm fertilizes an egg and form a zygote. You have cleavage that will divide that one cell to two cell, four cell, eight cell, etc. In terms of embryonic development. So there's two types of cleavage that can happen. Spiral cleavage is what protostome development goes with. It is where the planes of cell division are diagonal or vertical to each other, meaning that diagonal or vertical to the axis of the embryo. So the smaller cells actually center over larger underlying cells. They're not even size cells, and then they are not directly on top of each other where they're aligned perfectly with each other. Some actually have what's called determinate cleavage, where fate of each of these embryonic cells that you see here is set very early. So very early on, this cell is already determined to be a particular part of the organism. So if you were to just take a cell from this embryo, let's say a four-cell stage embryo, that one cell cannot develop into a whole animal because that cell is already determined to be a part of the body. Let's say that was for a hand or a tentacle. Okay. So if you take that cell can only become a tentacle. It cannot become a whole new organism. And that's what we mean by determinate cleavage. The fate of the cells are decided early on in development. Radial cleavage is then the other type of cleavage with deuteriostome development. That's when planes of cell division are parallel or perpendicular to the vertical axis of the embryo. And you can see here in this embryo that the cells are aligned perfectly on top of one directly above another, and that they are about similar size. Okay, that's what we mean by radial cleavage. So in these type of organ animals, usually they have what's called intermediate cleavage, where each cell, are produced by early cleavage, is able to develop into a complete embryo, the opposite of what we talked about with determinate cleavage here. So if you were to take a cell apart from the original embryo early on, that one cell can become the entire animal. And a good example of that in humans is in identical twins. So, identical twins are formed actually from the single embryo or zygote stage, where a sperm has fertilized the egg, and now you have this ball of cells that's maybe in the blastula stage. And all in all, it's just an early embryo where it can split into two balls of cells or two blastula in this case, and those two cells will then become individual human beings known as identical twins. They come from splitting of cells early on in pregnancy, usually not any later. Is usually happening, or we're talking about first trimester, very early on in the first trimester, that ball of cells can divide. Now, but how early it splits, sometimes the embryo can implant into the womb and then it splits within the uterine wall. That's when you can have identical twins that are in different amniotic sac and also supplied by different placenta, versus different sac but still sharing the same placenta, or identical twins that share the placenta and the amniotic sac, depending at what point did the early embryo start to split in development. But that's how identical twins are formed. So that's why for human beings, we have radial cleavage that are indeterminate as well, that is able to form a whole new human being if the cells were to, separate from each other early on. The second thing that is different between protostome and deuterostome development is the coelom formation. For coelom formation, remember that is the hollow space um, that the primitive gut that is forming within the air within the animal itself. So during gastrulation, here's the ectoderm, here's the endoderm, and then you have the red as the mesoderm. And the embryo's developing digestive tube is what we call this pouch right here called the archenteron. And coelom is then the pocket that is formed by the mesoderm. Okay, not the endoderm, but the mesoderm. So for protostome development, solid masses of mesoderm like this start to split from the endoderm, and then they form your coelom within there. See, that's why there's a hollow space inside the red cells. That's the coelom formed by the mesoderm. And then for deuterostome development, the mesoderm kind of buds from the wall of the archenteron itself, because here's the archenteron, and it starts to bud out like this, like a little the cells start to bud out, kind of like how exocytosis happens. And then, oops, and then you have mesoderm buds that is on top of this archenteron area, and then the cavity that is within that inside that blood is called the coelom. And third thing that can be different between protostome and deuterostome development is the fate of the blastopore. Remember, we said that when your primitive digestive tract is formed here, or the primitive gut is formed here, there is an opening at the top and opening at the bottom. And the bottom one is what we call the blastopore. So the indentation during gastrulation that leads to formation of the archenteron. So if you look back at this particular embryo, there's the archenteron, there's that opening at the bottom, that's the blastopore. Now, second opening usually form at the opposite end of the gastrula, where we had this and then this, so that there's an opening here and there's an opening there formed by that internal tube of the archenteron. So blastopore and that second opening becomes then the two openings of the digestive tube, which is your mouth and the anus. So for protostome development, the mouth develops from the blastopore itself, and that second opening at the top is then the anus. For deuteriostome development, the mouth develops from the second opening, where the blastopore then forms the anus. So it's just opposite in terms of which is the mouth, which is the anus. Okay, so again, for protostome development, the blastopore forms the mouth. Whereas for deuteriostome development, the blastopore becomes the anus, which means then the second opening up there is from the other thing, that the other opening. All right. So even though animals have lost through, where a lot of animal species were lost throughout mass extinctions on Earth, most animal phyla with members alive today were established around 500 million years ago, around that Cambrian explosion period we talked about. So zoologists currently recognize 36 phyla of living animals. That is, with 15 of them shown on this hypothesized phylogeny here in terms of how animals are related to each other. So bilaterians are divided into three main lineages. Here's bilaterias. Okay, so bilaterians are divided into your deuterostoma, Lithotrochozoa, and ecdysozoa. So for deuterostomia, these are hemichordates, which are, for example, acorn worms, your your echinoderms, okay, or echinodermata, which is then your starfishes, and then chordates, or chordata, which are then your vertebrates, and invertebrates, including humans, is in this particular phylum. It's a very, very diverse phylum we'll talk about in chapter 34. All right, and then the second lineage is the Lophotrochozoa right here, which forms again your ectoprocs, which is part of the Ectoprocta phyla, which has what's called a lophophore, which is a crown of ciliated tentacles like this for feeding purposes. So these are usually present in mollusk and annelids. We'll talk about those in the next chapter 33. And they have a distinctive developmental stage called the trophophore larva, which looks like this with little cilia that enable the larvae to swim. Hence, this is why this particular lineage is called Lophotrochozoa, come from the fact they have a lophophore and a trophophore larva. Third lineage is the ecdysozoa. So ecdysozoa includes your nematodes and your arthropods. So nematodes are worms like this, and arthropods or arthropoda is lobster, crustaceans, insects, etc. So for both of these species, they actually secrete external skeletons called exoskeletons, and then they will have to molt in order for an animal to grow, because that is a very hard shell on the outside. So in order for them to grow bigger in size, they actually have to shed off that exterior layer and allow the new layer from within to reveal. So that molting process is called ecdysis. Okay, ecdysis. All right, so here are the 5 key points of animal phylogeny, and we'll end with this for chapter 32. First of all, is that according to animal phylogeny, all animals share a common ancestor, as we see right here. Some ancestral protist is what they're thinking. And then sponges are actually sister groups. Sponges are represented by the phyla Porifera. So this is your basal taxon for the entire animal phylogeny. So they're sister group to all animals. Sponges are your basal animals, that basal taxon we talk about, and they diverge early from all other animals. Third, Eumetazoa is a group of animals with tissues. So that's why it's called usually means kind of like eudicots, true. And then this metazoa represents animals. That's why back then when we talk about dicots, we're talking about eudicots, true dicots. And this eumetozoa is then true animal because members of this group have tissues, like your muscular tissues and nervous tissues we talked about. Basal eumetozoan include your phyla Cnetophora and Cnidaria, which are both dipoblastic, and have radial symmetry. So if we go back to here, here's your Cnetophora and Cnidaria, okay, and eumetozoa. Fourth, most animal phyla belong to the group Bilateria. Bilateria. So bilateral symmetry is what they have, hence their name Bilateria. And they have three germ layers that are shared traits of this group, including your ectoderm, endoderm, and mesoderm. Majority of animal phyla in are animal that this particular phyla makes up the majority of the animal phyla in this particular group. Bilateria have the most amount of phyla. So here's all the phyla that falls under the group called Bilateria. So that's why Bilateria makes up the majority of the animal phylogeny. And lastly, you have three major groups of bilaterial animals. You have Deuteriostoma, Liphotrachozoa, and then Ecdysozoa. All have invertebrate, which are animals without a backbone. Chordata is the only phylum that includes vertebrates, or chordates is the only phylum that includes vertebrates, which are animals with a backbone, including us human beings. So remember, phylogenies are just hypotheses. They are not set in stone in textbooks and may change with new information and evidence. It is just something important to remember that science is always an ongoing dynamic process of inquiry. It will continue to change as new discoveries are made. So that's why textbooks have different editions, because they will change according to what science have newly discovered and confirmed. So that's all I have for you for chapter 32, an overview of animal diversity. We are at about 36 minutes or so. We still have practice questions, but be expected that chapter 33 and 34 are longer chapters. All right, so first practice question: what should animals with radial symmetry be better able to do than those with bilateral symmetry? Deals effectively with food distributed homogeneously in all directions. The key word there is all directions, because we talked about radial symmetry fits their particular lifestyle because they can't move. So it's nice if they can see their surroundings in all directions. Scientists researching the origins of multicellularity compared domains of blank proteins of unicellular organisms with those of certain animals. Cadherin, because we talked about that as one of the main key feature that allows for multicellularity, is how cells attach to each other and communicate with each other. That's the difference between single cell versus multi-cell animal, which is not a reason for the rise in the diversity of animal phyla during the Cambrian. Not a reason. We talked about three reasons. Global cooling triggers shifts in animal lineages. That was actually another reason for another era. So this was not for the Cambrian. The three hypotheses we talked about has to do with these other three other reasons here. All right. In which type of cleavage pattern does each cell have the capacity to develop into a complete embryo? Indeterminate cleavage, because that's the one where your fate of the cell is not determined early, so they have the capacity to become a complete embryo if they were to break off. Which taxonomic levels contains the other choices? Going with the phylogeny of animals, if you go back to that picture, you will see that Eumetozoa is the one that includes all the other ones, which is not true of the Neoproterozoic era. Not true. So something false. Animal diversity decrease, that is incorrect. If anything, that is when animal diversity start to increase, leading to your Ediacaran biota or Ediacaran period. What characterize an animal's body plan? All of the above. Symmetry, tissues, and body cavity, all that we talked about in this chapter. All right, place these groups in order in which they appeared according to the animal phylogeny. It's kind of funny. They put it in order. So it's 1, 2, 3, 4. Your Porphyra, Cnidaria, um, Acela, and then Nematoda, which is a member of Bilateria. Bilateria is right here, so which is a Nematoda. Porphyria is up here. Cnidaria is up here. Ctenophora, Ctenophora is up there. So that's why it's not. These three are not part of the Bilateria group. And that's all I have for you in chapter 32.